Good evening, dear students and respected teachers. Today we are going to discuss a topic by the name the complement system. May I have the first slide, please? Okay, myself, M.F. Mansoori. I am from Department of Microbiology, Srimati S.M. Panchal Science College, Talod. I have with me Professor Jagdish P. Patel. He is from Mehsana Urban Institute of Biotechnology, Kanpat University, Kanpat Vidyanagar. Now we are going to discuss, as I said before, the complement system. At the outset, I would like to thank the KCG team, the PISAC team, the SANDHAN team for providing us the opportunity of this lecture, and I'm grateful to them for giving us the opportunity. I'm also grateful to my principal, Dr. S.C. Parikh, for giving me this opportunity to deliver this talk. Let us see the contents of today's presentation. We'll begin with the introduction, that is, what is the complement system, its history, its definition, and the overview. Then we'll switch to the functions of the complement system. In this, there are many functions of the complement system which we will discuss. And then the components of the complement system. There are many components of the complement system and they all have a definite role. Then we'll discuss the pathways of activation of the complement system. There are mainly 3 pathways of activation of complement system which we'll see in this topic, which will be followed by the regulation of the complement system. Because complement system is so important, its regulation is also equally important. It helps us, but it may also damage us. So regulation is thus that important. Then we'll go to discuss biological consequences of this complement system. And finally, the complement deficiencies. What happens if there is a loss or absence of some components of the complement system. We'll begin our discussion with the history. Jules Bordet, who was an eminent scientist, he is considered to be a pioneer in the discovery of the complement system. He said that bacteriolytic activity requires 2 substances. First, the specific antibacterial antibodies, and the second, a heat-sensitive component responsible for lytic activity. He did his research with sheep anti-serum, and he found that the lysis of bacteria was not only because of the antibodies alone, but there was something else that would complete the action of antibodies, and that substance he named as a heat sensitive component. Later on, it was found that it was not a single component, but it was multi-component system. His research was followed by the research of Paul Ehrlich. We know his name. He is also the father of chemotherapy. He coined the term complement, and he said that this complement is some activity of blood serum that completes the action of antibody. You see, the completion of the work of the antibody is done by this complement system. By definition, we can say that a group of serum proteins, complement system is a group of serum proteins that participate in an enzymatic cascade, ultimately generating the cytolytic membrane attack complex. So it is many proteins that is participating in a cascade fashion, which will generate one complex known as membrane attack complex, which is cytolytic. We will hear this word membrane attack complex throughout our discussion, and in short it is also known as MAC or MAC. So researchers later on discovered that the action of complement was the result of interactions of a large and complex group of proteins. So it is the interaction between these proteins that will define the action. Now the functions of the complement system. There are more than 30 soluble and cell-bound proteins that are part of the complement system, and these proteins affect both innate and acquired immunity. Innate is something that we inherit, that is present in ourselves. Acquired is something that we develop during our life, and this complement system functions in both these immunities. Now, after it gets activated, after the complement system gets activated, it performs basic functions which are listed. The first function of the complement system is lysis, that is lysis of cells, bacteria, viruses and other microorganisms. The second function of this complement system is opsonization. You might have heard this word opsonization before. It is a system that precedes phagocytosis, that is, it promotes phagocytosis of particular antigen. It's like preparing a victim for. Opson is to prepare a victim for. The third function of the complement system is binding to specific receptors. Now these are many functions. This binding may trigger some specific cell functions like inflammation, secretion of immunoregulatory molecules, and many others. The final function of the complement system is immune clearance, means to clear the immune complexes from the circulation and deposit them in spleen and liver. What immune complexes? The complexes of antigen and antibody are immune complexes. When they are circulating, it is the function of this complement system to clear them from the circulation and bring them to the spleen and liver for the destruction. So we can see pictorially the functions, the various functions. The first function, that is lysis. As you can see that the complement acts on the target cell. It brings about the lysis of this target cell. The opsonization is engulfing, the step before the engulfment of the bacteria by phagocytes. As you can see that complement receptors are present in the phagocyte, and the complement, the orange triangular structures which are present on the bacteria, they bind, and that is how the opsonization takes place. The third function is like many functions, triggering specific cell functions, activating the inflammatory response, which includes degranulation, extravasation. Degranulation is like removing the granules or moving the content of a cell out of the cell, that is degranulation. Or extravasation is, vasation as you know is related to vascular system, and that is bringing something out of the vascular system is extravasation. You see that the blood which is flowing in the capillaries, and the cells from this blood are removed from these capillaries into the tissues, that is extravasation. That is part of the inflammatory response. The final function, that is immune clearance, is visible in this slide. That is, the antigen antibody complexes, many complexes, are removed from the circulation by the phagocytes and deposited into the spleen and the liver. Now let us discuss the components of the complement system. The components of the complement system are proteins and glycoproteins. Now who synthesize these components? Our own cells synthesize these components, and many cells synthesize these components. The cells that synthesize these components are liver hepatocytes, blood monocytes, tissue macrophages, epithelial cells of the gastrointestinal and the genitourinary tract. All these cells synthesize the complement components. Now most of these components, the complement proteins, they circulate in serum in an inactive form known as proenzyme. Pro means something before, proenzyme, or zymogens. Zymogens are inactive forms, and this word is commonly used for enzymes. These are made active, these zymogens, these proenzymes are made active by proteolytic cleavage. This cleavage cuts the protein, cuts these glycoproteins and activates them, the cutting bringing two fragments, one large fragment, one small fragment. So after this cleavage, after this cut, the smaller fragment is designated as A and the larger fragment is designated as B. For example, C3 is a component of the complement and it is broken down into C3A and C3B. Similarly, C4 may be a component which can be broken down into C4A and C4B. But here C2 is an exception, means whereas A is the smaller component, but C2A is the larger component, but that is only just in C2, that is C2A is a large cleavage fragment. As you can see that C3 is broken down into C3B, C3A, the C3B is the larger component, C3A is the smaller component. The larger fragment binds to the target site of activation. It performs the main function of the complement system. And the smaller fragment is not like functionless. It is not like throwing away into the dustbin. It also has some definite function. It diffuses from that site and it can initiate localized inflammatory response by binding to specific receptors. So the larger has a greater role. The smaller doesn't mean that it has a smaller role, but it has a role away from that site and it is smaller in size. As you can see in this slide that the C4 component is broken down into C4A and C4B. The C4B stays there at that site and the C4A leaves that site. Similarly, in case of C2, which is broken down into C2A and the C2B. C2A is the larger component which stays at that site and C2B leaves that site and performs some other localized inflammatory function. Now these complement fragments, they interact with one another to form functional complexes. Means there is binding between the C4s and the C2 components. These complexes have enzymatic activity and they are designated by a bar over the number or by some symbol. Example, the C4B2A, that is two components C4B and C2A. That complex is represented by a bar which is shown below. Similarly, C3B, the factor B small component B is also a complex, and there is a third complex known as C4B 2A 3B complex. Now, the word cascade. The complement reaction sequence starts with an enzyme cascade. You might be aware that cascade is like many events unfolding one after the other, where the first event of that sequence triggers another component of the second of the next part. So one stimulates the other, the second one stimulates the third one stimulates the fourth one. This is what is the meaning of a cascade. Now, complement components are designated by numbers or by symbols or by names. Like complement components are numbered like C1, C2 and so on till C9. They are also represented by factor D, factor B, or by names like properdin or homologous restriction factor. Now, coming to the next topic of discussion, that is complement activation. So the first steps, the initial steps, the early steps occur by one of the three pathways. The early steps result in the formation of the fragment C5b. So these early steps culminating, that is resulting in the formation of C5b can occur by the classical pathway, the alternate pathway, or the lectin pathway. This is one shot of how the alternate pathway, the classical pathway converge to finally form C5 through C3. The final steps after the early steps, final steps that lead to a membrane attack are the same in all the pathways. So whether the complement pathway is initiated by the first pathway, that is the classical pathway, or the alternate pathway, or the lectin pathway, all of them ultimately result in the final steps, that is membrane attack, which is common to all the pathways. Let us discuss the first pathway, the classical pathway. The classical pathway begins when an antibody binds to an antigen, that is antigen-antibody complex forms. Now this complex stimulates the C1 component of this pathway, that is C1 gets activated by binding to the antigen-antibody complex. Now this is the visual of C1 component. C1 component is again a multi-subcomponent. It has C1q, which is visible as the arms, the finger-like projections, the heads and the spirals. The C1r and C1s are the globular structures, the ring-like globes that are visible. So C1q, C1r and C1s form the whole C1 complex. The formation of the antigen-antibody complex induces a change in the Fc portion of the IgM molecule, that is the antibody molecule, that expose a binding site for the C1 component of the complement system. That is, when antigen is bound by an antibody like an IgM, there is some change taking place in the IgM, the Fc portion of the IgM. Now this conformational change, this change induces the binding of the C1 component to that antigen-antibody complex. So the